All right, we're going to cover background about chemical reactions and then get more in depth about enzymes and then talk about what pH is and acids and bases. All right, so chemical reaction is a process where you're going to rearrange or like break bonds, form bonds. You're going to rearrange and join atoms together to form like compounds and molecules. When you have a chemical reaction, mass and energy are conserved. I mean, you're not going to lose or gain any mass or energy. Again, you're just rearranging your atoms, how they're joined together. You're going to maybe break some bonds in one compound, break some bonds in another compound, and rearrange their atoms to form new compounds. So the things that are going into reaction are called reactants, while the elements or compounds that come out of the chemical reaction are known as products. So just an example here, just for in a biology sense, in your bloodstream, you have a chemical reaction happening where you have carbon dioxide. Well, we'll talk about it in the next slide, but carbon dioxide and how you breathe that in or how you breathe it out is a process of a chemical reaction. So carbon dioxide reacts with water in your blood to produce carbonic acid, which is very soluble. So this chemical reaction enables blood to carry carbon dioxide to the lungs. And that's kind of a way that your body tells if it needs to breathe in and out based on the pH of your blood. So this carbonic acid, when it's acidic, your body knows, oh, I need to breathe out this carbon dioxide. So in the lungs, the reaction is reversed. So you have carbonic acid being broken down here, being broken down into carbon dioxide and water. So the carbon dioxide moves to your lungs, so then you can breathe it out, and the water will retain in your blood. So this is just an example. It's not something you're going to be tested on, but just an example of how chemical reactions happen within your body. So we have many different chemical reactions, but this is just an example of one. So now talking about energy in chemical reactions, determining how if a reaction will happen or not is based on something called activation energy, which we'll talk about in a little bit. So energy changes. Energy is released or absorbed whenever chemical bonds are formed. So you're going to release energy when you break a bond, absorb energy when you form a bond. Energy changes are one of the most important factors in determining whether a chemical reaction will actually happen. So chemical reactions that release energy often will occur on their own or known as spontaneously, while chemical reactions that would need to absorb energy won't really happen unless they have some source of energy. So for example, if you burn hydrogen gas, which hydrogen gas is very flammable, so burning happens in the presence of oxygen gas. When you burn hydrogen gas, you'll get an explosion and form water. So the explosion is the energy that's being released. So this would happen spontaneously if you expose hydrogen gas to a spark and oxygen, you'll get this explosion, which is the release of energy from this reaction. So again, just an example, not something you're going to be tested on. The reverse reaction, if you had water and you wanted to separate the hydrogen and oxygen, you need a lot of energy to do that. So it won't happen spontaneous. You're not going to see a puddle of water just break apart into hydrogen and oxygen. So you can do that if you pass like an electrical current through it. That would be your energy source that would decompose water into its hydrogen gas and oxygen. So and only in one direction here the reaction produces energy. In the other direction, the reaction would require energy. So again, you're not going to be tested on this, but just an example of a reaction needing energy and a reaction that would happen on its own and produce energy. So energy sources. So for chemical reactions, every organism has to have some sort of source of energy to carry out the chemical reactions that happen within us for metabolism purposes to stay alive. So plants get their energy from the sun through photosynthesis. Animals or heterotrophs, also another name, need to consume their energy, get their energy from consuming plants or animals. So when we're looking at chemical reactions that release energy do not always occur spontaneously. The energy that's needed to get a reaction to actually start or to happen is something called activation energy. So that's going to kind of dictate whether a reaction will happen on its own or not. So these are two graphs of the activation energy needed for reactions, an absorbing reaction and a releasing reaction. The peak of each graph represents the energy need for the reaction to go forward. So they both have pretty much the same, maybe this one has a little higher, activation energy, okay? The difference between the required energy, so the peak, and the energy of the reactants, so for this absorbing reaction, here's your reactant energy, and the activation energy, so, or the energy needed for the reaction to happen. So this difference here is the activation energy. Same idea here for the energy releasing reaction. The reactants energy need is here, or the energy the reactants are bringing in is here. The energy required for the reaction to happen is here, so that difference here is the activation energy, okay? Um, so just seeing the difference between absorbing and releasing, you can tell that this reaction based on the curve is absorbing energy because the reactants are at a lower energy level than the products. So if your product's energy is higher than your reactants, it means that sometime during this reaction, it absorbed energy. For it to tell if you're working with an energy releasing reaction, because this you're going to see graphs on the test, and it'll be good to know when this is an absorbing reaction because the products are a higher energy than the reactants. This one is a releasing energy one because the products are at a lower energy than the reactants. So if your reactant energy is higher than the product energy, it means at some point during this reaction you lost energy. So that's a way to tell if you are working with a energy releasing reaction. If your products are lower than your reactants, then you released energy. All right, now specifically about enzymes, we talked a little bit about enzymes during digestion, but now we're going to kind of go more, talk a little more detail about enzymes in general. So some chemical reactions that happen in your body are too slow or have activation energies that are too high that would make them practical for living tissue. Like the reactions wouldn't happen because they're too slow, or they won't have enough energy absorption for activation energy. So these types of chemical reactions are made possible by the use of catalysts. So a catalyst is a substance that's going to help speed up the rate of a chemical reaction, and how they do that is by they lower the activation energy that's required to start a reaction. So in a biological sense, a biological catalyst we refer to as an enzyme. These are made out of proteins. So again, referring back to macromolecules there. Within a living being, enzymes, which are biological catalysts, speed up chemical reactions that would take place in cells. So again, they lower activation energies, which makes these reactions very quick and more efficient for any sort of process that needs to happen within the body very quickly. So for example, again, just to kind of tie back an example, when the reaction in which carbon dioxide combines with water to produce carbonic acid, it's really slow. So it's so slow that carbon dioxide might build up in the body faster than the bloodstream could actually remove it. So your body's going to use an enzyme called carbonic anhydrase to actually speed up this reaction by a factor of 10 million, so that the reaction is going to take place pretty much immediately, and carbon dioxide is going to be removed from the blood as soon as it's pretty much present. So you need this to happen quickly so that you get rid of your carbon dioxide, and so that your cells then you later can breathe in oxygen, so your cells can use oxygen for something called cellular respiration. So you need to remove this carbon dioxide fast, and this is what the enzyme carbonic anhydrase allows your body to do. Otherwise, you wouldn't be able to release it fast enough for your cells to actually live. So enzymes are actually very, very specific, generally catalyzing only one chemical reaction. So like carbonic anhydrase only works with carbonic acid, okay? Part of an enzyme's name usually derives from the reaction it catalyzes. So yeah, again, the carbonic anhydrase only is going to work with carbonic acid, no other different compound. When you're looking at an enzyme, the reactions of enzymes, the enzyme catalyst reactions are known as substrates. So a substrate is anything that an enzyme will work on, okay? So for example, the enzyme carbonic anhydrase converts the substrates, carbon dioxide and water into carbonic acid. So whatever the enzyme is working with, so for example, amylase in your mouth works specifically with starch, okay? It won't work with other things like protein or lipids, but it works specifically with starch. The reason why is that the substrate, so here's like a substrate, binds to a site on the enzyme called the activation site. So here's a enzyme, big blue object here. Here's the activation site. This activation site on the enzyme is specific for this substrate. So they often kind of use the analogy of a key being specific for one lock, okay? So there's a very big specificity with an enzyme and the substrate it works with. Okay, so they're kind of like soulmates in a way, or like a key, a specific key that would fit into a specific lock. So enzymes can be regulated based on the temperature they're at and the pH. So certain enzymes work better at specific temperatures, and there's enzymes that have specific pHs that they work better at. So enzymes that are in your stomach work better at lower acidic pHs, whereas enzymes in your small intestine, large intestine work better at pH values that are more neutral or maybe slightly basic. So we're going to talk about pH right now and what exactly that means. So pH scale, it ranges from 0 to 14. It's a measurement of how many hydrogen ions are in your solution, which we'll talk about why that's important. At a pH of 7, the concentration of hydrogen ions and hydroxide ions are equal. So like water has equal hydrogen and hydroxide, H2O. So equal parts hydrogen and hydroxide, so it has a neutral pH of 7. Acids form hydrogen in your solution. So when you have a lot of hydrogen in your solution, you're an acid. And that would be equivalent to a pH of below 7. So the closer you are to 1, the stronger you are. So like strong acids have a pH of like 1 to 3. So here, stomach acid, pretty potent, a pH of 1 to like 2 here. So here, 0 to like right before 7 are things that are acidic. So some acid rain, tomato juice, lemon juice. If you ever had a cut in your mouth that burns, so that's acidic. Vice versa, a base is something that produces hydroxide, OH ions, and they have a low concentration of hydrogen. So what that would be equivalent to is a pH value above 7. So strong bases are closer to like 14, so range from 11 to 14. So here, above 7 here, this blue area, whitish to blue area, are things that are bases. So bases tend to be kind of like from everyday items would be like cleaners, anything that would kind of wash areas of your house. Acidic things tend to be more acidic or more edible. Okay. Buffers. Okay, a buffer, very important for homeostasis. The pH of the fluids within most cells in the human body must generally be kept between 6.5 and 7.5, a neutral. So if the pH is lower or higher, it'll affect the chemical reactions that take place. So one of the ways that pH is controlled is the use of a buffer, which is a weak acid or base that prevents a sharp, sudden change in pH and keeps the pH within a certain limit. So just as an example here, you want to just know pretty much the term buffer, but to see how it works, here if we had a base that was not buffered, poured a strong acid in it, it would turn acidic. Here though, if we have a base that was buffered, pour a strong acid in there, this buffer prevents the remaining solution to turn acidic. So it might become a little slightly lower pH or acidic, but the overall pH remains overall basic. So a buffer prevents any sort of strong change in pH if you add a strong acid or base. Okay, that is it. Thank you.